Welcome, welcome. We are going to now focus on one of the kind of most important things in biology. In fact, it's so important. It's called Eukaryotic central dogma, meaning this is like the foundation. This is literally why we have such a great diversity in terms of the different cells in our body, the different the animals on the planet. And it all starts with our DNA. And eukaryotic central dogma actually includes two separate processes. It's taking DNA and converting it into RNA, and then taking that RNA and converting it into all of these different proteins, which carry out all of the many functions in our body and contribute greatly to the diversity and the array of different types of cells that we have as these very multicellular eukaryotic organisms. So the DNA to RNA process is a process known as transcription. So I like to think of it as languages, right? If you think of DNA and RNA, they are both nucleic acids. So they are in the language of nucleotides. Transcription is going from the same language and kind of taking it down. So if you were working as a transcriber, say in a court proceeding, you would be writing down everything that the people were saying, and you'd be writing it down. If they were speaking in English, you'd be writing it in English. So that's the same language. So that's the transcription process, going from DNA to RNA, because it's still the language of nucleotides. Going from DNA to RNA is translation. So now we're going into a different language. We're going from the language of nucleic acids and nucleotides to the language of amino acids and proteins. Think of sitting in the UN in New York City, the General Assembly, and somebody is speaking in one language and they have a translator who is then translating into hundreds of other languages. Because we're changing a language, nucleotides to amino acids, then that's a translation process. Students often mix those two processes up, but if you think of it in terms of language, I think it's kind of easier to keep them separate. So we start, of course, with the DNA. And if you think of DNA, you can basically think of DNA as kind of the master blueprint, right? That's what we're going to do. We're going to build something. Well, in order to build something, we have to have a blueprint. But DNA is all of the blueprints. It has plans and ideas for every possible protein that your body can make. And that type of information is contained in a gene. It's a segment of a DNA strand that has the instructions for one type of polypeptide. So if you think of all of your DNA as all of the plans, a gene is just one plan. That gene has both what we call exons and introns. The exons are the parts of the DNA that actually code and have information for amino acids. The introns are these non-coding segments, and actually may even include viral DNA. We have some viral DNA in ours, which is kind of interesting. Our genetic library works on what we call triplets. So each DNA triplet, 3 nucleotide sequence in the DNA, tells us the number of amino acids, the kind of amino acids that we need, and the order in which they should appear. And when we put these triplets together in a gene, we can then transcribe, i.e. kind of take that information down and turn it into RNA. And then once we have it in RNA, we can then translate it into the actual sequence of amino acids. So in order for this process to occur, we have to use not only our DNA, bound up in our nucleus, but then all 3 types of RNA that we find in the body. The mRNA, or messenger RNA, which is, if you think of the DNA as the kind of master blueprint, say the blueprint for the whole house, mRNA is then the blueprint for a single window of the house. It's the instructions that are going to kind of be carried from the DNA to the kind of building protein. Ribosomal RNA is a structural component of our ribosomes, so it helps the ribosomes kind of interpret the mRNA, and then transfer RNA is what actually kind of carries the amino acids. It kind of picks them up and brings them over to the ribosome in order for them to be attached into our amino acid sequence. So our 2 processes, again, are transcription and translation. Transcription takes place in the nucleus because DNA cannot leave the nucleus. We are going to transcribe the DNA into messenger RNA. That messenger RNA then kind of slips out the little nuclear pores out into the cytosol, which is where translation takes place. It will involve our little ribosome here, whether it's a free ribosome like this or perhaps one that's attached to endoplasmic reticulum. And that is where we will build our polypeptide, and we will build our protein. So DNA to mRNA, same language, transcription. mRNA to proteins, different language, translation. This is what transcription looks like. This is the DNA to RNA process. So let's zoom in. So very similar to the DNA replication, we still have to unwind it, right, because it's bound up into the helix. And essentially what occurs, so kind of a 3-step process. We start with a little promoter region that's like, oh, this is where we want to start. This is where our gene starts. We have 2 different strands of DNA, right? We have our coding strand and our template strand. And then elongation is the process of adding amino acid, no, excuse me, not amino acid, ribosomal with the mRNA. I'm having a, goodness gracious, my brain died. Anybody ever have that happen where you're talking and then something's on the tip of your tongue and it just flies out your head? That's what just happened to me. So elongation is adding the nucleotides of the mRNA into a nice little strand. And then lastly, we're going to end with this little termination signal, and that is when our little RNA polymerase, we always have enzymes that help us do things, kind of splits off. The DNA will rewind, and we have our mRNA strand. So let's talk about coding strands versus template strands. And to do that, we're going to come over here to the bigger picture. So let's say we have a particular piece of DNA, and that piece of DNA is AGC TAC. I just randomly picked a couple nucleotides, but genes are usually much longer than that, but we're going to just work with 2 triplets just for the sake of kind of ease. Well, that 2 sets of triplets, AGC, TAC, that is the information that we want. That is the code, the instructions that we want. So we want an mRNA molecule that looks just like that one, that has the exact same sequence, A, G, C, U, A, C. Because remember, we're talking about mRNA, we're talking about RNA, and there are no thymine nucleotides. We have the uracil nucleotides. But A, G, C, T, A, C, that's the DNA. A, G, C, U, A, C, that's the mRNA. And that is the same exact information. That's our coding strand. But because polymerase enzymes work on base pairing, in order to get the coding strand into mRNA, in order for our mRNA to look just like our coding strand, the strand of DNA that we use as a template to base pair against is actually the other strand of DNA. So this other strand is then our template. So if we, again, want AGC-TAC, that's what we want, but we want it in mRNA format, we're going to use the opposite piece of DNA as our template. That opposite piece of DNA would be TCG, ATG. And then if we base paired to get our mRNA, we would end up with AGC-UAC, base pairing off of the template strand. So that is why we have both a coding strand for our gene and a template strand. The coding strand is the information we want, and in order to get that information, we have to use the template strand in order to base pair properly. Once we have our mRNA, the mRNA will leave the nucleus, and then we're going to translate that mRNA into a sequence of amino acids. So let's take it out of the cell for instance again. Let's go think, actually not biology again. If you think of DNA as the blueprint for your house, you're going to build a house, and the mRNA is the plan for one window in the house. The tRNA is then all of your laborers. It's all your little worker bees going to pick up the bricks and pick up the wood and bring it over to the construction site to build the window in the house. Where that is occurring, the construction site, is then the ribosome with the ribosomal RNA attached. And those are our three types of RNA. We got the mRNA, that's the blueprint for the one window, the tRNA, the little worker bee, and the rRNA with the ribosome, which is the construction site. When it comes to our genetic code in our messenger RNA, the DNA triplets are then transcribed into what we call mRNA codons. And the mRNA codon actually then contains all of the information for the amino acids. That's what actually codes for the different amino acids. And you can see a lot of amino acids are represented by more than one codon. The amino acid alanine, any of these four codons will result in saying, hey, I should have an alanine amino acid here, and so on and so forth. AUG is our start codon. It also codes for the amino acid methionine, so every single polypeptide in our body actually starts with a methionine amino acid, and then there are three different codons that basically say, okay, stop, end, we're done. This is the end of the gene that we want. And so that way we know we can stop there and we can stop building our polypeptide. So this is what our tRNA looks like. It actually has two different regions, kind of this stem region here, which is where the amino acid kind of gets grabbed onto, and then a head region down here, which has an anticodon to help it bind to an mRNA codon. Now the anticodon doesn't contain genetic information. It's basically a backstop, so that if our codon, our plan, calls for GCG, which if we go back, GCG, we get GCG, that codes for the amino acid alanine. if we want alanine, we have to make sure that we get alanine. And if we don't get that, we're going to get the wrong amino acid, we're going to get the wrong sequence, and screwy things can happen. And so the anticodons are basically a way to make sure that we get the correct amino acid when that amino acid is called for. It's not genetic information. It's just a way of kind of making sure and verifying. It's a backstop, a backup plan. So this is what the ribosome looks like. It actually consists of 2 subunits plus the rRNA. And in order for translation to start, the big, large ribosomal subunit and the small ribosomal subunit, and a little initiator tRNA here, which is going to have that methionine amino acid attached to it, comes and binds onto the 2 ribosomal subunits along with the messenger RNA. That kind of starts us off building our protein. The ribosome itself actually has 3 different sites: an A site, a P site, and an E site. The A site is where the new amino acid will kind of attach onto this. The P site is where we're going to form a bond between the one amino acid and the next amino acid, and then the E site is when the kind of sequence of amino acids kind of gets to detach from our ribosome. So let's take a look at what that process looks like. Once we have initiated our process, then we can get elongation occurring, and it's all about recognitions of the codons. Codons are the information because they're the mRNA, they're the plans. So when the codon comes into the A site, that is then kind of recognized, and we say, okay, it's CUA, that means we need leucine. Well, a tRNA with the complementary anticodon, so that we for sure get leucine, comes and binds onto this particular A site. We get a peptide bond forming between the amino acid attached to this tRNA and the amino acid attached to this tRNA, and then everything shifts over, and then this tRNA can be released, and we repeat. A amino acid, P for the peptide bond, E for the exit, and this goes on and on. Amino acid, bond, exit, shift, 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 as our strand of amino acids gets longer and longer and longer. And then eventually our stop codon will end up in that A site. We'll get a release factor, and the amino acid chain will be released from the ribosome. The 2 subunits will break apart, releasing the messenger RNA, and now we have a fully formed polypeptide chain, which can then fold into its specific conformation. And that is the process of translation. This is a very nice summary figure of the process of eukaryotic central dogma, transcription and translation. So you can see here is our DNA triplets off of our one gene. So if you think of DNA as the plans for the whole house, one gene is the plan for one part of that house. We transcribe those triplets into mRNA codons. The mRNA codons have the information for the amino acids, and the anticodons on the tRNA help us make sure that we get the proper amino acids that are called for by our codons. The last thing that I want to discuss here is what we call cell differentiation. And again, every cell in your body, with the exceptions of the ones that are anucleate, have the same DNA. Doesn't matter if it's a nervous cell, a fat cell, a muscle cell, a take your pick cell. As long as this particular cell has a nucleus, it has the same DNA as every other cell that has a nucleus. But all our cells are really, really different, right? A fat cell doesn't look anything like a muscle cell, which doesn't look anything like a nervous system cell. The reason we have such a great variety in terms of our cellular structure and function is this differentiation process. Essentially what happens is when we are in those early embryonic stages, different cells get different chemical signals, they get different contact signals, and that kind of sets them on these very specific pathways by activating and deactivating different genes. And by activating and deactivating different genes, that ensures that some types of proteins are made and other types of proteins are not. And by changing the kind of types of proteins that are made, cells are then able to specialize to all of their very unique and specific functions that we see in our adult cells. And none of that would be possible without transcription and translation, which is why it is eukaryotic central dogma.